Genomes of model organisms. A model organism is a non-human species that is extensively studied to understand particular biological phenomena, with the expectation that discoveries made in the organism model will provide insight into the workings of other organisms. Model organisms are in vivo models and are widely used to research human disease when human experimentation would be unfeasible or unethical. This strategy is made possible by the common descent of all living organisms and the conservation of metabolic and developmental pathways and genetic material over the course of evolution. Studying model organisms can be informative, but care must be taken when extrapolating from one organism to another. In researching human disease, model organisms allow for better understanding the disease process without the added risk of harming an actual human. The species chosen will usually meet a determined taxonomic equivalency to humans, so as to react to disease or its treatment in a way that resembles human physiology as needed. Although biological activity in a model organism does not ensure an effect in humans, many drugs, treatments and cures for human diseases are developed in part with the guidance of animal models. There are three main types of disease models: homologous, isomorphic and predictive. Homologous animals have the same causes, symptoms and treatment options as would humans who have the same disease. Isomorphic animals share the same symptoms and treatments. Predictive models are similar to particular human disease in only a couple of aspects, but are useful in isolating and making predictions about the mechanism of a set of disease features. Model organisms are chosen on the basis that they are amenable to experimental manipulation. This usually will include characteristics such as short life cycle, techniques for genetic manipulation such as inbred strains, stem cell lines and methods of transformation, and non-specialist living requirements. Sometimes the genome arrangement facilitates the sequencing of the model organism's genome, for example, by being very compact or having a low proportion of junk DNA, for example yeast, Arabidopsis or pufferfish. When researchers look for an organism to use in their studies, they look for several traits. Among these are size, generation time, accessibility, manipulation, genetics, conservation of mechanisms and potential economic benefit. As comparative molecular biology has become more common, some researchers have sought model organisms from a wide assortment of lineages on the tree of life. Important model organisms. Model organisms are drawn from all three domains of life as well as viruses. The most widely studied prokaryotic model organism is Escherichia coli or E. coli, which has been intensively investigated for over 60 years. It is a common gram-negative gut bacterium which can be grown and cultured easily and inexpensively in a laboratory setting. It is the most widely used organism in molecular genetics and is an important species in the field of biotechnology and microbiology, where it has served as the host organism for the majority of work with recombinant DNA. Simple model eukaryotes include Baker's yeast such as Saccharomyces cerevisiae and fission yeast such as Saccharomyces bombi, both of which share many characters with higher cells, including those of humans. For instance, many cell division genes that are critical for the development of cancer have been discovered in yeast. Chlamydomonas reinhardti, a unicellular green algae with well studied genetics, is used to study photosynthesis and motility. C. reinhardti has many known and mapped mutants and expressed sequence tags, and there are advanced methods for genetic transformation and selection of genes. Dictyostelium discoideum is used in molecular biology and genetics and is studied as an example of cell communication, differentiation and programmed cell death. Among invertebrates, the fruit fly Drosophila melanogaster is famous as a subject of genetics experiments by Thomas Hunt Morgan and others. They are easily raised in the lab with rapid generations, high fecundity, few chromosomes and easily induced observable mutations. The nematode Caenorhabditis elegans is used for understanding the genetic control of development and physiology. It was first proposed as a model for neuronal development by Sidney Brenner in 1963 and has been extensively used in many different contexts since then. Caenorhabditis elegans was the first multicellular organism whose genome was completely sequenced and as of 2012 the only organisms to have its connectome or neuronal wiring diagram completed. Arabidopsis thaliana is currently the most popular model plant. Its small stature and short generation time facilitates rapid genetic studies and many phenotypic and biochemical mutants have been mapped. Arabidopsis was the first plant to have its genome sequenced. Among vertebrates, guinea pigs, Caviar porcellus were used by Robert Koch and other early bacteriologists as a host for bacterial infections, becoming a byword for laboratory animal, but are less commonly used today. The classic model vertebrate is currently the mouse, Mus musculus. Many inbred strains exist as well as lines selected for particular traits, often of medical interest, for example, body size, obesity, muscularity and voluntary wheel running behavior. Among vertebrates, the rat, Rattus norvegicus, is particularly useful as a toxicology model and as a neurological model and source of primary cell cultures owing to the larger size of organs and suborganellar structures relative to the mouse, while eggs and embryos from Xenopus tropicalis and Xenopus levis or the African clawed frog are used in developmental biology, cell biology, toxicology and neuroscience. Likewise, the zebrafish Danio rario has a nearly transparent body during early development, which provides unique visual access to the animal's internal anatomy during this time period. Zebrafish are used to study development, toxicology and toxipathology, specific gene functions and roles of signaling pathways. Other important model organisms and some of their uses include T4 phage or viral infection, Tetrahymena thermophylla for intracellular processes, maize for studying transposons, hydras for regeneration and morphogenesis, cats for neurophysiology, chickens for development, dogs for respiratory and cardiovascular systems, Northobranchus furzeri for aging and non-human primates such as the rhesus macaque and chimpanzee for hepatitis, HIV, Parkinson's disease, cognition and vaccines. Ensemble is a joint scientific project between the European Bioinformatics Institute and the Wellcome Trust Sanger Institute, which was launched in 1999 in response to the imminent completion of the Human Genome Project. After 10 years in existence, Ensemble's aim remains to provide a centralized resource for geneticists, molecular biologists and other researchers studying the genomes of our own species and other vertebrates and model organisms. Ensemble is one of the several well-known genome browsers for the retrieval of genomic information. Similar databases and browsers are found at NCBI and the University of California, Santa Cruz, UCSC. This is a screenshot of Ensemble database. UCSC Genome Browser. The UCSC Genome Browser is an online genome browser hosted by the University of California, Santa Cruz, UCSC. It is an interactive website offering access to genome sequence data from a variety of vertebrate and invertebrate species and major model organisms, integrated with a large collection of aligned annotations. The browser is a graphical viewer optimized to support fast interactive performance and is an open source web based tool suite built on top of a MySQL database for rapid visualization, examination and querying of the data at many levels. The Genome Browser database browsing tools, downloadable data files and documentation can all be found on the UCSC Genome Bioinformatics website. This is a screenshot of the UCSC Genome browser.